Point pattern analysis studies the spatial arrangement of points in a geographical space. It has applications in a wide range of areas, including wildlife ecology, biology, epidemiology, natural resource management, and criminology. For analysts, it is often relevant to not only know that point-event clustering occurs, but also where the clustering occurs. This presentation outlines several available approaches to identify the location of point clusters. The primary focus will be the kernel density estimation approach. Besides explaining the principle of kernel density estimation and the role of kernel density functions, the different options that are available to scale the values of grid cells in a kernel density map will be demonstrated. Clusters occur in a geographic distribution when features are found in close proximity or when groups of features with similarly high or low values are found together. The latter are denoted as hotspots and cold spots, respectively. Cluster location identification is useful to be able to take action based upon the location of one or more clusters. Two examples include the assignment of a police task force to deal with a cluster of burglaries or intensifying treatment efforts for pest control managers in observed clusters of insect pests. This presentation and the next one will discuss methods to detect clusters that are based on spatial proximity between events. Numerous cluster detection and visualization techniques are currently available, including kernel density estimation, nearest neighbor hierarchical clustering, and scan statistics. This presentation focuses on kernel density estimation, whereas nearest neighbor hierarchical clustering and scan statistics will be discussed in the next presentation of this point pattern analysis series. Kernel density estimation is an exploratory method to identify the location of clusters as areas of high local event densities. The concept is that the event pattern has a density at any location in the study region and not just at locations where there is an event. This principle results in continuous surfaces of density estimates. This figure provides an example of a 3D visualization of a kernel density map. Higher peaks indicate areas of a higher local density of events compared to other local regions. Kernel density estimation methods have a variety of applications, including exploratory point data analysis, point data smoothing, and creation of continuous surfaces from point data in order to combine or compare these with other datasets that are continuous. Kernel density estimation methods estimate the event density by counting the number of events in a region. This region is called a kernel and is centered at the location where the estimate is to be made. Kernel density functions are used to weight nearby events more heavily than distant ones in estimating the local density. Many different kernel density functions exist. They are symmetric functions centered at 0 with an area underneath that equals to 1. This figure shows some common univariate kernel density functions. They are called univariate since their values vary only along one axis. Most of the kernel density functions are bounded, which means that they count only events within a given threshold distance from the location where the density estimate is made. This threshold distance is called the bandwidth. Accordingly, the units along the horizontal axis of kernel density functions are multiples of bandwidths. Examples for bounded kernel density functions are the uniform function and the quartic function, as indicated by the yellow arrows. As opposed to bounded kernel density functions with unbounded kernel density functions, each event in the study region contributes to the estimated event density at a given location. This means that these kernel density functions reach beyond the specified bandwidth. An example of this category is the Gaussian or normal kernel density function, which is commonly used in kernel density estimations. Kernel density functions can be extended to 2 dimensions. A bivariate kernel density function is created by effectively rotating the univariate probability density function around the z-axis going through the center point of the univariate probability density function. As an illustration of this concept, this figure shows a bivariate Gaussian kernel density function that is centered at 0 and has a standard deviation of 1. It is called a bivariate kernel density function since the kernel density value varies along 2 coordinate axes and not only one. Estimating the density value at a reference location starts with placing the 2-dimensional kernel function over each event point in the study region and calculating the density value contributed by that point over a grid. This is illustrated to the left, where red dots indicate the location of events, and the bumps show the contribution of each event to the estimated grid cells and areas surrounding an event. The yellow star shows a reference location for which an estimated density value is sought. Values for all event points are then added together for each grid cell to give a composite density surface. This allows the estimated density value for each grid location, such as the selected reference location shown to the right, to be read. The composite density surface can then be plotted in 2 dimensions. For example, the plots can show density contours, a colored grid map, or a 3D surface. The data grid values that result from the kernel density estimation may be scaled in 3 different ways. This will be illustrated with a simplified dataset of 105 deer sighting locations shown in this figure. In this dataset, the point spacing between nearest sighting locations is 50 meters. Each dot corresponds to 1 sighting. The first way to scale the resulting data grid values is to provide an estimated number of events per grid cell. These are referred to as absolute densities. With this method, the sum of the values across all cells should equal the number of events used in the analysis. The software used to compute grid values shown in this map was CrimeStat. A normal kernel function with a bandwidth of 100 meters was applied. The data was rendered in ArcGIS, which provides kernel density estimation procedures as well. The legend shows that the estimated number of events per grid cell varies between 0 and 0.84. The statistics of the grid cells to the right indicates that the sum of the values across all cells is approximately 105. This corresponds to the number of deer sightings in the study area as expected. The second way to provide grid values is as relative densities. These provide values in events per unit area. They are adjusted by the grid size, such as events per square meter, hectare, or square mile. Event density per square mile was used to generate this map. The legend shows that the estimated event density varies between 0 and about 872 events per square mile. The third way to provide grid values is as probabilities. Probabilities are computed as absolute densities divided by the total number of events in the study area. In such a map, a grid value shows which proportion of all events is located in a specific cell. The legend shows that cells with the highest event density contain about 0.8% of all events in the study area. With this method, the sum of the values across all cells equals 1. This can be seen by the summary statistics of cell values to the right. This slide summarizes the presentation. It presented one of several commonly used methods of point cluster detection, namely kernel density estimation. It explained the different types of univariate kernel density functions and their role in kernel density estimation. Lastly, the presentation illustrated how to obtain different map units through scaling of grid cell values.